Okay, welcome back. This is an exciting series of lectures, for me at least, because this is where we really get to integrate. So I've told you all along that physiology is really about capturing information across scales, bringing it all together and describing some macroscopic phenomenon, maybe even some clinical phenomena. And we're really getting to that stage now. We've been working our way through from the cell to the tissue, and now we're going to talk about the whole heart. So stay tuned for a little bit of insight into how the heart works electrically. So as usual, we start with our theory section. So this is where I give you some of the background on how this all works. And I'm fortunate enough to have a video that I could find to explain it to you, so I don't have to make a fantastic one. Okay, we'll walk you through this video, and it's really meant to highlight the various elements, structural elements, as usual, we're going from structure to function again, that arise in the heart and exist in the heart and really give it this integrated function. So we'll see there are cardiac cells that have special purpose. You've heard of some of these before. We'll try and tie that all together and really explain how a heartbeat happens. Now, the beginning of this whole process starts at the sinoatrial node. That's a special bundle of tissue. We've talked about it a little bit before. This is where rate control of the heart really happens under natural circumstances, where the autonomic nervous system can influence the rate of heart. The sinoatrial node stimulates, sends excitation through the atria, and that stimulation moves as a wave, as we've described in our tissue context, and reaches another special node or group of cells called the atrioventricular node, which has a lot of complexity and structure within it. But from a macroscopic perspective, its main purpose is to couple the excitation from the atria to the ventricles, but with a delay. There's an internal delay. This is the only place in a normal heart where there's electrical coupling between the atria and the ventricles. Once that excitation passes through the AV node and the AV bundle, then it reaches the bundle of His, which is a very rapidly, very speedy highway of conduction that then splits into subbundal branches, the left and right, and they move down both sides of the ventricles beneath the tissues on the subendocardium. And then that excitation spreads through a Purkinje fiber network, it's called, and reaches the entire ventricles. Now this second stage, this phase through the Purkinje fibers is very rapid. It moves very, very quickly, and you can see the consequences of all of this would be that we'd have a delay in the activation from the atria to the ventricles, which, as a consequence, creates a time for blood to flow from the atria into the ventricles. And then this rapid conduction through the Purkinje fibers causes a synchronized contraction of the entire ventricle in a very short time span. And it also determines the sequence in which that starts. So it generally starts from the apex, that's the part at the bottom, and then moves up to the base of the heart. And you can imagine how that squeezes in this sort of a twisting motion it has. We'll come to the mechanics of it very shortly. But you can imagine how in that just that sequence of contraction will tend to squeeze the blood out of the ventricles. And that is indeed what happens. Now, as we'll get to in a little bit, this whole process has electrical consequences. And we'll see that encapsulated in the ECG, which is one of the things that we will spend a lot more time talking about and actually using in the lab. So here's another image, a still image, that captures all these various specialized tissues. This is called the conduction system in the heart. And you can see here the sinoatrial node, the pathways through the atria. There's lots of really ongoing research about what those pathways are and how they work. And you can see also that the speed of propagation, we talked about this last time, the spread of activation in the tissue, the propagation velocity is quite variable across these different regions. And you see orders of magnitude difference between, for example, the conduction through the AV node at 0.05 meters per second up to something on the order of 1 or 2 meters per second in the bundles, the His bundle and the Purkinje fibers themselves. So there's a lot of specialized tissue here. We could spend lots of time describing how that all happens at the cellular level, at the tissue level, but we're really interested in the whole heart level at this point. So we can characterize each one of these features. Now this is an image, a graph, whatever, that I also like a lot because it really describes the whole sequence of events, because each one of those red tracings is schematic, but it characterizes different action potential shapes in different parts of the heart, and time separates them as they, more or less as they happen, again, schematically, in the actual spread of activation in the heart, in a heartbeat. So the first thing to excite, of course, the sinoatrial node, and then you can follow it down through those same elements of the conduction system that we've just seen in animation and in a diagram. Now the ECG tracing at the bottom is nothing more complicated than the sum of that entire process. And of course it's kind of ridiculously complicated, and we'll get into a little bit of the pieces of that, but you can perhaps at least qualitatively grasp how each one of these electrically active regions of the heart will contribute some signals, some current, that eventually we can record in an ECG. And now we'll dive a little bit into how that process works and how we should think about that. There's another figure before we go to that brings the two ideas together. So it's kind of a localized version of that previous image with the action potentials from different parts of the heart, and then a schematic anatomical drawing of the heart that shows where these different types of action potentials arise, and again with the ECG at the bottom. That will hopefully help you put kind of time and space and these different action potentials into some sort of picture of a sequence of events with a lot of specialized tissues involved. Now these are all muscle cells, these are not nerve cells. These are all bundles or nodes of cardiac muscle. Some of them are not contractile, strangely enough. The Purkinje fibers do not contract. That's one of their really important features that I think we've talked about. And so there's variety across each one of those tissues and gives them their specialized function. Here's another sequence of images that, again, in still, sort of discrete steps walk us through this. We have the depolarization from the sinoatrial node, this spreading across the atria. You see the purple region is the active region. It eventually reaches the entire left and right atria, both the atria, and then encounters the AV node. It passes slowly through that node and then rapidly depolarizes through the Purkinje system. And then that depolarization wave, that excitation wave, that then moves from the apex up toward the base of the heart. So here again is another rendering of this sequence. There are many, many more if you're curious. The internet seems to be full of them. Now the next step in this process is to picture how we get from the heart to the body surface, because the body surface is where electrocardiograms are measured. And we'll put that together in theory, and then in a subsequent video get a little bit into, well, a lot into the actual measurements, which we'll do in the lab this week. So we have several components we have to talk about. So the first one is what we call a source, a bioelectric source. Every time we move to another scale, we have to come up with another underlying model that describes the behavior of interest. We cannot picture an entire heart as the sum of 10 billion individual cells. It just defies certainly our brain capacity and our computing capacity. So we have to use models at a different scale. And we've talked a little bit about that already in the tissue scale, and we'll see more of that. So that's the source model part. And there are a number of different source models, just to make things interesting and confusing. Then you have a source model, let's say, something generating current that's sitting inside what we call a volume conductor. The thorax is our volume conductor, and it has its own complexities because it has complex shape. It has different tissues, different organs within that thorax, and each one of those has variable electrical conductivity. Electrical conductivity that in some cases can even depend on direction, and that's called anisotropic conductivity. So this is passive conductivity of the tissues through which this current has to flow to get to the body surface to make an ECG. And then finally we get the whole question we'll talk about in the next video of how we measure, what sort of electrodes we have and where we put them and how do we interpret those signals. So the origins of our source model, one of them at least, goes back to a diagram we spent some time with in the previous videos, when we described how tissue conducts this spread of activation. And at a small scale, 2-cell scale, this is the picture we have. Now it's those extracellular currents again that are the source. That's the bioelectric field or current source that we will be trying to measure, trying to localize, trying to interpret. Seen at a slightly coarser scale, we can picture kind of this block of tissue and we have current flowing inside the tissue, current flowing outside the tissue. We've seen these kind of images before. Here's yet another rendering showing this at an even bigger scale where there's multiple cells here, more than just 2 cells, with a boundary between these regions that are resting and regions that are activated. And that's spread in this case from left to right, from the activated tissue spreading, moving into the resting domain. And that leads to some source formulations that we can picture. Now, the whole idea of these is always to find a simpler rendering, a simpler conceptual model for each one of these underlying complex models. The simplest one, which we will see used in many, many different ways, and certainly is the core of a lot of clinical electrocardiography, is this notion of what's called a dipole, a current dipole. A current dipole you can think of in the simplest sense of being a region that is a source of current and a region that is a sink for that current closely brought together. In the ideal physical manifestation, both of them are sitting in the same space, and so they have no physical domain and all these wonderful things that work in physics and math, but not so well in physiology. But we can picture this being a reasonable surrogate to say that, okay, we have a region that's activated, it's the source of intracellular current. We have a region that's at rest, it absorbs that intracellular current, and then we have the reverse on the outside. So the outside, it looks like there's current flowing backwards from the resting region to the excited or depolarized region. And that we can picture as a little battery and hence a dipole source. Now we can picture it just as a single strand of tissue with one dipole, and that dipole is moving, just to make things interesting. So we have a moving dipole along that series. But we can also get a little higher scale still. Remember when we looked at those spreading waves of excitation in tissue, we saw they formed elliptical shapes. And so this is really a three-dimensional structure. So now we can picture a three-dimensional region that's excited, and at the boundaries around that region, we have a number of individual dipoles. We can picture this discretized so that not just one dipole, but each little segment of that boundary between the excited and the resting tissue becomes its own little dipole. And that becomes a dipole source that we can also kind of capture. Then we can get even more anatomically realistic and even bigger scale where we have this cross-section, very schematic again, cross-section through the ventricles. And now we see this purple region is excited and the white region is at rest. And at each boundary between the excited and the resting region, we could picture a dipole. In this case, 3 dipoles capturing this wavefront as it moves up the left and right and septal regions of the ventricles. Now, all these are actually used for various types of study in various settings. But the one that ultimately matters most clinically is, believe it or not, a single dipole, where we represent that entire complex activity of the heart as a single moving dipole, moving in space and certainly moving in orientation as time goes on. And so this is really the heart dipole vector that is the fundamental notion, the fundamental source notion behind a lot of electrocardiography, practical electrocardiography, like the clinicians practice in the hospitals. And here we see another rendering of that sequence of events, in this case, just the ventricles. So here the dark region is the region being excited, and then the gray region is the region still at rest. And you see at the top of this figure, an initial activation, in this case in the septum between the 2 ventricles, and it's moving from beneath the left ventricular wall towards the right ventricle. And you can see there's a little arrow there that shows that activation. And that sequence then moves on as we go from top to bottom through various stages of the spreading activation. And at every moment in time, all that activity is sort of summarized into one single dipole that, again, is moving in space, changing in orientation. As you can also see in the length of that arrow is changing in amplitude. It turns out that that's a wonderfully simple source that we can actually capture and render and even localize in space from measurements we make further away. So further away could be outside the heart, could be on the body surface, could be the electrocardiogram. And that's what you see in the other two columns of this figure. You see the vector at that moment in time extracted from the anatomical space and plopped inside this triangle. This is an important triangle. This is the triangle that Eindhoven created to capture this behavior. And the electrodes that make up this, the most basic, what we call limb leads, they sit on the apex or the corners of this triangle. And they represent locations where we place electrodes, make measurements. The measurement is the difference in the voltages between those 2 points. And so it works like a projection. So now we're in a vector space that we should be comfortable with, in which we project that vector onto axes. And those axes, the signals on those axes, then become the reflection of that vector inside the triangle. Now this is a weird coordinate system. There's no question. We're used to seeing X and Y. If we were engineers, X, Y, maybe Z if we were in three-dimensional space. This is two-dimensional. It's a triangle. Why in the world would you do that? There are reasons for it that we don't have a lot of time to get into, but the point of the matter is it doesn't actually matter mathematically. We can have coordinate systems that are orthogonal, but we can also have coordinate systems that take any angles you want. As long as we have two axes, they don't have to be orthogonal in mathematical terms. And so here we have a triangular space. We have three individual nodes, so three electrodes, and then we subtract the signals from those three electrodes in three different ways, and then we get three different projections. Now what's in that middle column is exactly a rendering of that. Each one of those black arrows on the sides of the triangle corresponds to a projection of the vector in the middle of the triangle to each one of those three directions, on each one of those three sides. And we can see as the vector changes its orientation and amplitude, so too do the projections change. Now on the right-hand side, what you see is the time signal associated with each one of those projections. And the projections are labeled here with Roman numerals, 1, 2, 3. That's this standard nomenclature. Don't let it bother you. It's not because this goes back to Roman times. It's not that old. But it was developed by a Dutch physiologist and physician named Eindhoven, who won a Nobel Prize for this again. It was a very, very huge breakthrough. We'll get to more details of that history in the next video. But the point here, understanding the theory here, is that we have a vector quantity that we're projecting onto an unusual coordinate system from which we can then reproduce the vector direction from those recordings. So the idea here is not necessarily to go from left to right, because if we have the information in the left column, we have everything we need to know. The idea more often is to say from the recordings we can make on the body surface, which are cheap and painless and easy to make, we want to be able to move back to the heart itself and use that to intuit what electrical activity is going on in the heart, within the heart at that time. And that's really the big picture of all of these electrocardiographic approaches, is to try and understand those relationships. So now that's a little bit about sources. Now we'll get into volume conductors. So again, this source, the heart, the dipole, sitting inside a region, the thorax, that has variable conductivity and variable shape and all sorts of other confusing things. So here's a simple schematic, again, two-dimensional slice through the thorax in which we see a number of different tissues, each with their own conductivities or resistivities of the units that this is captured in. The blood has a very low resistivity or high conductivity. That is probably somewhat intuitive. The bone, on the other hand, is at the other extreme. It has a very high resistivity or low conductivity. And each different region has its own conductivity. If we're going to understand the way the signal reaches the surface of the body, then we have to incorporate in our thinking and eventually in our modeling what those tissues are, what shape they have, what conductivity they have, and that's indeed what happens. We now being locally here at the University of Utah, have actually created tools to generate these volume conductor models from, let's say, MRI scans. And here you see a rendering again of a thorax scanned from a human. This is a human subject. And then you see different colors representing the different regions that we identified. It's called segmentation, this identification of different regions that correspond to different conductivities. And then you see three different views of that rendering, that segmentation that we've performed. Other people do the same thing. We get models that, in this case, for example, contain, like you saw, a dozen different tissues, all with different conductivities. And here they're, again, just captured and rendered somewhat artistically, but beautifully, showing a thorax with all the various tissues, the bone, the lungs, the heart, all the internal organs, that sort of thing. So this is how we capture the behavior of the volume conductor and try and accommodate for it when we interpret the ECG signals. At this stage, of our education, I think all we need to know is that there is a volume conductor, it is complicated, and of course it will, if nothing else, weaken the signal. So the source, the signal coming from the heart, will have a larger amplitude, but just because it spreads out, just like a light source, by the time it gets to the body surface, it will have reduced amplitude. And that's something we also have to take into consideration when it comes to measuring this. Now, we put all this together, we put a source together with a volume conductor and then imagine measurements. And then we have what I'm going to show you in this video, a beautifully simple but quite accurate and realistic rendering of the currents as they flow. Now what you see in this video is, it's a very old video, it literally dates back to the 60s, but it captures very well this merging together of the active heart in the light colored regions in the middle, the currents that flow from that activated region through the rest of the heart, through the rest of the thorax, and then proceed to run along the edges of the thorax itself. Now the thorax contains all the electricity. The electricity doesn't leak out. It doesn't cross into the air that surrounds the thorax, but we can measure that passing of current along the edge of the body as the ECG. And that's really the core theoretical idea behind the ECG that we'll see and learn a lot about measuring. And that really is the core of the underlying theory here. So think source model, again, of which there are many, embedded in a volume conductor, which is complex, three-dimensional, potentially anisotropic, all that wonderful stuff. Those two together can generate a voltage, a small voltage, that we can sample on the body surface, and that's the ECG that we'll talk about in a few minutes in the next video. Thanks for your attention. Bye-bye for now.